welcome to Generation Hope, the interview series, a behind-the-scenes companion to our upcoming documentary, Generation Hope. This series features full-length, unfiltered conversations with the scientists, doctors, researchers, and advocates who are on the cutting edge of gene therapy, as well as the families living with rare genetic disorders like Phelan-McDermid syndrome, Rett syndrome, STX-BP1, and Angelman syndrome. While the documentary follows the personal journeys of children and their families navigating these conditions, this interview series offers a deeper dive into the science, the hope, and the human stories driving this historic moment in medicine. We call this project Generation Hope because we believe this generation of children could be the last to suffer from these devastating genetic disorders. With advancements underway, we believe this generation will mark a turning point, a moment in history when everything began to change. This generation shall be known as Generation Hope. Gene therapy is no longer a fantasy. It's a reality that's unfolding in real time. The question is no longer if, it's when. We created this series to share that hope, to show you what's coming, and to remind every parent, every family, and every person affected by these disorders that you're not alone, and the future may be brighter than you've ever dared to imagine. Thank you for joining us on this journey of resilience, discovery, and hope. Dr. Alexander Kolevzon is a highly esteemed child and adolescent psychiatrist, serving as the clinical director of the Seaver Autism Center for Research and Treatment and professor of psychiatry and pediatrics at the Icahn School of Medicine at Mount Sinai. A leading voice in the field, Dr. Kolevzon has received numerous awards for his contributions to advancing research and treatment for neurodevelopmental disorders, including the Rare Champion of Hope Award. He has worked tirelessly within the Phelan-McDermid syndrome community, spearheading clinical trials and driving research that has provided critical insights into understanding and treating PMS. Today, we're honored to hear from Dr. Kolevzon about his groundbreaking work and the hope it offers to families around the world. So I thought we'd talk about kind of three things. One, I want to talk about you, kind of your background from, when you were a little kid and what inspired you to kind of get into this.
Little kid.
Origin story.
Yeah.
And then I want to talk about, I'm also in town for the Icons conference. So I want to get kind of your thoughts on that and then gene therapy. And everything that kind of involves kind of around there.
Yeah.
So I'm going to start off with, tell me about little Al.
Origin story. Well, let's see. We are sitting now in Mount Sinai Hospital, and it's 2024. I was born across the street in Mount Sinai Hospital. I won't say the date. It was a while ago. I grew up here in New York City, on the Upper West Side. Let's see. I mean, there's many iterations. I guess I went to college to be a psychiatrist. That was my intention. I was sort of drawn to the field. I did terribly in college. So I dropped pre-med. I was a psychology major in the end. And then I went back to school for 2 more years to kind of catch up and do all my pre-med requirements. And then I went to medical school. But again, with the intention to be a psychiatrist. Loved psychiatry, loved pediatrics, and sort of debated a bit between those two fields and chose to work with child psychiatry. Before college, right at the beginning of college, I think I was the summer after my freshman year, I was a camp counselor in a local camp here at 92nd Street Y for kids with autism. That was the first time I ever met a child with autism. And I was really fascinated by it. I was struck by how impaired these kids were, but at the same time, many of them had these remarkable gifts. And it was just a very kind of interesting experience for me. And it sort of planted a seed. I had no idea that I'd go on to kind of pursue, working in this area. Graduated from medical school, came here for residency. So this is sort of full circle for me, and fellowship. And kind of midway through my residency, was encouraged, invited, persuaded to pursue clinical research. And at the time, there was already a well-established autism center here at Mount Sinai, which I became very involved in. There's a lot of background noise.
Believe it or not, I think AI will be able to get rid of that.
Oh, that's cool.
Yeah,
Nice.
it's insane.
It's amazing.
Yeah, I know.
I started using like ChatGPT now for.
Oh, yeah. Oh, it's good. That's one of the things I want to talk to you about.
Yeah, I don't know much about it from a research perspective, but from like a life perspective, instead of asking Google, I'll ask ChatGPT. It's amazing.
Yeah.
So I had always expected to be a clinician and go into private practice. Really, that was kind of the model that I had developed in my mind anyway. And when I was invited to do research, I got really kind of fascinated by asking these questions and writing papers and analyzing data. But because there was an autism center here, and because I had that experience as a camp counselor, when I was asked, hey, what do you want to do in terms of research? I was given sort of a menu of options, essentially, and autism was among them. And I said, oh, autism, yeah, I had that experience, many years ago. And that was 2002, 2003. Mm-hmm. And then, kind of, ever since then, I've been pursuing the idea of developing better treatments. Sometime around 2008, 2009, I realized we were doing a terrible job of it, mostly because autism was so heterogeneous biologically. So the idea of trying to apply one treatment to a group of kids that were so different was not especially effective. And that's when we started to think about specific genetic forms of autism. And SHANK3 was, the number one at the time kind of gene of interest for us as a center. And really not a lot of people were working in this area.
And this is 2008.
This is 2008, 2009. Yeah, I need to actually nail down that exact date because I've been asked for it. But certainly by 2010, we were sort of in full swing. I think Joe Buxbaum, who's the director of our center, had published the first paper out of our group in a mouse model of SHANK3 out of our group. And then we started to test various treatments in that mouse model and then applied those treatments that worked in the mice to kids.
What was the IGF?
Exactly. Insulin-like growth factor 1. Exactly. Yeah. And I'm still working on it 15 years later. I'm still working on that today.
I think that was the first paper I read on this.
Oh, yeah.
And it was shortly after it was published because we got Evie's diagnosis in 2016.
Yeah. I think that first paper was published in 2014.
Yeah.
And we had started to kind of develop the idea in 2010 after having seen some patients. Yeah.
I was curious about how you feel about the idea of there still being a categorization of autism, because it is I've got a personal feeling about it as kind of it's dated and needs to be put away because now we have genetic genetic diagnoses.
Yeah.
It's like someone going to a hospital and saying, yeah, they're sick. You're sick with a bunch of different things. But
fever, right. Yeah. I mean, we criticize our own diagnostic manual, right? It's kind of the diagnostic and statistical manual of mental disorders is really a sort of unknown disease at the moment. And every time a specific cause gets identified, the disorder kind of drops out of the manual, Rett syndrome is a perfect example of that. In DSM-4, Rett syndrome was a diagnosis under autism. And it is no longer. Fragile X syndrome doesn't exist.
That's interesting in itself because in Canada, a lot of the funding that parents get is because the child gets an autism diagnosis. And if they don't get an autism diagnosis, the funding's not there.
Yeah.
So it's,
Very, very true in the States also. Very, very true. Yeah. So, autism is still a behavioral diagnosis. I think it has like some clear meaning. And, more than 50% of the cases, we don't know what the gene is, right? We can't identify a gene in, more than 50% of the cases. We still know it's kind of genetic in origin, but we don't really know exactly what those genes are. I think with more testing and sort of more knowledge and more advanced technology and analytic methods and so on, we'll start to identify more genes. And then, more and more, it will become kind of meaningless. You'll just sort of say, oh, they've got a collection of symptoms, but it's really due to this specific gene.
Right, that's DXP1. Yeah.
But there are still kids that have this kind of classic autism, the same kind of features that Connor described in the 40s, where you can kind of see them from across the room. And, they may not have any other organ system involvement. They may not have any clear dysmorphic features. You do genetic testing and you see nothing of significance. So, there's still going to be an autism left even after we sort of learn more and more about the genetics, I think.
Mm-hmm.
Yeah. So I would keep it in the DSM. I would sort of have these kind of qualifiers, right? Autism due to SHANK3, and that exists.
Right.
It's now kind of a specific line that you fill out, basically.
So you guys started working on SHANK3 because that was at the time kind of one of the known sort of genetic disorders that had a autism phenotype, or?
Yeah, so it was definitely identified as an autism gene. It was not well researched from a clinical perspective, meaning kind of patients being characterized. I think there were 400 cases known in the world at the time.
Mm-hmm.
So yeah, again, 2008-ish. And not a lot of people were working on it. So we thought of it as kind of a good opportunity to learn more. And this is sort of following along the lines of Fragile X syndrome and Rett syndrome and so on. So yeah, we saw SHANK3 as the next really, really important gene that caused autism.
Mm-hmm.
And the first studies would estimate that like 85% of kids, including studies of ours, 85% of kids have autism. But then over time, we learn that they don't really have autism in the kind of more rigorous sense of the diagnosis, closer to 60%.
60% of kids?
Of kids with Phelan-McDermid syndrome will meet criteria for autism, kind of as best as we can apply them to this population.
Right.
It's still, you talk to most parents and they'll say, they carry an autism diagnosis. It's not really autism. And, the parents are usually right.
Yeah. Yeah. It's, it's, the behaviors kind of are separate from, the genetic diagnosis, and, two kids with the same, like, I've met kids with Phelan-McDermid syndrome who are fully verbal and are, can toilet themselves, can feed themselves, and all these things. And there's and with the same kind of single point mutation. I remember the first girl I met, first Phelan-McDermid syndrome patient that I met, and that I saw. I was having conversation with her, and I was blew my mind.
Yeah,
because my daughter is not like that.
Right. It blows my mind. And I've seen lots of cases where the identical sequence variant can present totally differently in two different kids. It's mysterious. But back to the autism issue, there are kids with Phelan-McDermid syndrome who are very clearly autistic in the kind of classic sense. And then there are kids who are very clearly not. And that kind of spans, different developmental levels as well. But yeah, I think it's strategically important, and I've said this a lot to families, to keep SHANK3 connected to autism because it's familiar. People understand what that is. It's a pathway to get better services.
Yeah.
It's a pathway to get more research funding. The whole natural history study and the development synaptopathies consortium was sort of founded around the idea that these are autism genes, even if they don't cause autism in every case.
What about? I was curious if regression, because regression is really common in Phelan-McDermid syndrome, and if the patients with PMS who never had a regression, if there's a correlation between those patients who don't show signs of autism versus if someone had a regression, if they all show signs of autism.
Yeah, so I don't think we've seen a higher rate of autism among people who regress. And I also wouldn't necessarily say that that regression is common. It's we're identifying it more and more, but like among the entire universe, people with Phelan-McDermid syndrome, may not be more than 30 or 40%.
Okay.
And there's different regressions, right? There's sort of regression that occurs at the younger ages, 3, 4, 5, 6, and then around puberty. The pubertal regression is potentially more dramatic and not characteristic of sort of, what we call idiopathic autism. But, kids with idiopathic autism, they'll have regressions, and, probably about 30% of them, but those are earlier regressions, 3, 4, that kind of thing. But you're asking about ways to potentially predict in advance who might be at risk of regression. We don't know the answer to that. I think some of it probably has to do with baseline levels of functioning and language.
Right,
I mean,
sure.
quite obviously, if you have language to begin with, you're more likely to be able to see loss of language, and if you don't have language, you might not. But we don't know a ton of that, other than potentially the fact that smaller deletion sequence variants in particular are at higher risk. But those are also kids that are exactly yeah. So it's very complicated. We also know a little bit about what the triggers are in terms of regression, right? So it could be infection, it could be major life changes, it could be traumatic events. Whether those are causal or correlated, we don't really know yet.
Yeah. So one of the things that or essentially that I'm here in New York for is the International Consortium on Newborn Sequencing. So there are a bunch of studies going on around the U.S. Where they are offering whole genome sequencing to basically any baby or any child that ends up in the NICU for any reason, or is behind any of their milestones for any reason.
Yeah.
And the hope is to offer kind of whole genome sequencing early on, so that families don't have to kind of search forever. For our family, it took 4 years. From when Evie was, just 5 years, from when Evie was just about 18 months, and we started seeing her fall behind on milestones. And she was nonverbal till she was about 4, and she started having some words, and then at 4.5 to 5, how old is she now? She's 14, just turned 14. At 4.5 to 5, she had a regression in terms of she was very social, and she would play, and she would have some words, and she would feed herself fine, and she was toilet trained. And then she lost all of it. And not just that, but she just retreated. She started just licking windows, stemming nonstop, and she developed pica. And to this, she's got most of those things to this day.
Yeah.
But it took, and so we went through that entire thing not knowing what's going on.
Oh, I see. So was it the regression that triggered the need for genetic testing?
Yes.
I see.
Yeah, because before that, she was just kind of behind on all her milestones. She was global developmental delay. And then the regression hit, and from then it still took 2 years. So it took basically 4 or 5 years.
Does she have a sequence variant?
Yeah.
Okay.
Yeah, she has single point mutation there.
So the sort of standard array didn't pick it up.
That's right.
Right. Exactly.
Yeah.
And you're in Canada. Yeah.
So at first they did the array, didn't pick it up. And then, and that took, I think, 6 months or so to get the results back. And then they did the exome with my wife and I and Evie, and then it came back. Phelan-McDermid syndrome.
Yeah.
So that whole diagnostic odyssey that we were on, and a lot of families are on, and don't know what's going on in their child, and they're thinking all kinds of terrible things that could be happening to them at school, or that we don't know about, and diet and all these things. And so there's the emotional effect on family, and then there's a fiscal and financial effect on the system. How many neurologists did we go to who had no idea what was going on, and metabolic specialists, and nobody knew what was going on.
Yeah.
So the whole idea of the early diagnosis is, what is this going to save the system, and what is it, what is it going to mean for families?
Mm-hmm.
Do you see any reason why, any ethical reasons or anything, why whole genome sequencing early on, were there any issues with it, or?
So, I'm obviously a proponent of genetic testing. I think more information is valuable. I think eventually we'll have a much better sense of, being able to predict disease course and intervene early, and even think about prevention of certain features. But a lot of it has to do with people's personal beliefs. And I think not everybody is interested in all that information. Even now, there's lots of information that's available if you ask for it. And But often people don't want to know. The question I always get asked is, what does this mean in terms of, manipulations? Because if you can test at birth and you can test prenatally, and are we going to start to, select babies based on genetic changes? So I think people do express concerns about it. From like a medical scientific perspective, I think it's a really important thing to be doing. But I certainly hear a lot of arguments for the contrary. I mean, theoretical arguments. And then I talked to a lot of families where, they're coming with developmental delays or autism even, clearly diagnosed, and they don't necessarily want to do genetic testing. Well, yeah, I think that's not uncommon. Usually when you talk about the potential benefits, I think they understand more, but I don't know. I mean, it's, it's different philosophies for different people.
Right.
Yeah.
So the newborn screening panel is something that's done in like the pinprick for every baby, and that's kind of scans for 50. Right now it's 50.
50 things that are
50 disorders.
Mostly where there's actually like an intervention.
Only when there's an intervention. So the latest thing that was added was SMA.
Yeah.
Because of Dunsper.
For sure.
So I guess, if things go well with the Jaguar gene therapy trials, or with the Taysha Rett syndrome trials, or I think Angelman. Is that also getting a gene therapy trial soon?
ASO, yeah. They reported some pilot data from their ASO trial. Yeah.
ASO?
Antisense oligonucleotide. So it's a way of sort of upregulating the expression of the intact allele.
Is that done with, what's that? Is that done with, it's not gene therapy, is it?
Well, so it's a genetic manipulation, but you're not inserting a gene.
Right.
But it is potentially disease modifying. Right? So with SHANK3, you've got two alleles, right? One's got a healthy copy, one's got an absent, basically, SHANK3. Mm-hmm. But if you can attach a molecule to the healthy copy and ramp up expression, you could potentially be replacing the chain or the deficiency.
Wow. So this is kind of why I'm doing this documentary.
Yeah.
I feel like we're on like the cusp of, exactly, of a new world, a world that has never existed before.
Yeah.
And having, specifically like, kids with, or not just kids, but, people with like intellectual disabilities, kind of their brains kind of coming online at all different ages.
Mm-hmm.
And have you sat back and kind of played through how this is going to go and what that means?
I mean, I'm still frankly reeling from the fact that we're where we are right now, to be honest. I mean, you talk about neonatal screening. I think once there are more treatments available, people will be much more likely to want screening for obvious reasons. But when we started in this space, we were just thinking about small molecules that were, hopefully going to treat some of the symptoms, but never imagined that, 10 years, 15 years forward, we'd be actually doing gene therapy trials. We've been burned a lot. There's been lots of promise over the years that has not borne out.
Sure.
So I'm generally cautiously optimistic.
Yeah.
And I'm kind of focused on where we are right now and making sure the trials that we're preparing for are going to be safe. So I haven't quite played out the kind of best case version of this. But yeah, it's easy to imagine, right? This being life-changing for lots of families and for the whole population, really.
When I spoke with Gooping, we did an interview in Minnesota. Going to get a tour of his lab on Friday.
Nice. Yeah.
And to him, it was, I was very encouraged to hear some, the kind of the tone in which he was. It wasn't a question of if.
Oh, sure.
It's just a question of when.
I agree with that.
And so, and I felt that way when Evie got her diagnosis.
Yeah.
In 2016.
Yeah.
The 2018 conference in, was it Phoenix, Houston, Texas, somewhere in Texas, for the Phelan-McDermid syndrome conference.
Probably Orlando.
No, it was the other one. I think it was the second one.
Okay.
It was. I arrived. It was the first time we knew we just got a diagnosis, and I flown down, and I'd arrived to everyone frantically running around because one of the kids went missing.
Right.
They weren't missing. They were hiding under a table. But as soon as I got there, I just dropped my bag and went out to the 100-degree weather outside in the parking lot looking for, stuff.
Yeah. But
I remember at that conference, nobody was talking about gene therapy.
Yeah.
And I had asked several of the researchers that were there, and they all looked at me, kind of, yeah, we'll get there someday. 20, 30 years, we'll get there.
Yeah.
And like you said, it's 8 years later and we're at, human trials.
I know.
It's amazing. So to me, it's also the same. It's just a question of when, not if.
I think it's absolutely a question of when, not if. Absolutely.
So I've been going through that exercise of, and that's why I'm doing this documentary. Because what does that mean? What does that mean for my daughter? What does that mean for someone who's 30 years old?
Yeah.
They're 50, 60-year-old people with Phelan-McDermid syndrome, and their parents are still taking care of them.
Yeah.
And it is, when I tell the story to people, they think of, there's this one movie that you probably know with Robert De Niro, or
Yes.
Awakenings. And this is Awakenings on a global scale.
Yeah.
Have you stopped to like actually?
Yeah. So, the question I get asked a lot is the impact in younger kids versus older kids, potentially,
Yeah.
So I think, Awakenings is a very dramatic example, but those are folks that sort of developed typically, right? And then kind of got locked in.
Mm-hmm.
I think for an adult with Phelan-McDermid syndrome who's lived a life of developmentally being so delayed, it could mean learning some profoundly important new skills, which can be life-changing for families. But I don't think we're going to kind of rescue a typical development it seems like less likely to me that they're going to, start, talking and doing advanced math. But I think that, as you know, even the tiniest of benefits can be really impactful for families. So if you can start to teach some new skills, if you can start to teach some communication, I think if SHANK3 is replaced in essence, and you've got, intact nerve cell connections, it would be absolutely amazing. Absolutely amazing.
Be thinking of this cohort of people, whether, with all kinds of different syndromes that are going to have experiences that nobody else on earth has ever experienced and been able to share, and what that means for them, and who are they going to become? Like, very empathetic people, right?
Yeah.
And how and what their contribution is going to be to society, because we're going to learn so much from them. And then there's the question of, what are they going to remember? Are they going to remember everything? Are they not going to really remember anything? Is it going to be like the first 2 years of life where you don't really remember much their first 2 years?
Comes to what I was picturing as you were describing it.
Yeah. So these are all unknowns that we're all, going to find out over the coming years, I guess.
Yeah, I think if you fast forward 5 years and we actually have an approved treatment that's truly disease modifying, and we're doing screening for everybody, you won't. It'd be a good thing, right? You won't know. You'll just drop the gene in there as soon as the diagnosis occurs, and people are not going to remember what life was like before. But it's. To me, just the kind of promise of it, just the hope. It helps families so much. It helps the scientists so much. And even if the first treatment sort of out of the gate is not just a slam dunk, there's so many companies that are interested, right? These companies are all kind of jumping on and recognizing that SHANK3 is such an important gene. And there's so many different ways of tackling this that something is going to work for sure. Again, back to, it's not if, it's when. Yeah.
So you're familiar with Rett syndrome, obviously. And are you, have you ever received funding from the Rett Syndrome Research Trust?
I haven't done any real work in Rett.
So I mean, a lot of the funding that they've raised over, I think, $70 million.
70.
Over the past 10 years. And they've funded a lot of research. They've got, I think, 3 trials going on now, or 2 and 1 starting.
3 gene therapy trials.
Yeah. So I think 2 they have ongoing. They're about I think they're about a year ahead of Jaguar.
Okay. But they have a first in human. They have an AAV9 trial.
Yeah. That one was done in Montreal.
Yeah.
Oddly enough. So I was asking Jaguar if they're also doing it in Montreal, but no, they're not.
Yeah.
And they have so that one's Taysha, and then they have Neurogene, which is I forget if it's also. I don't think it's also AAV9. But they're funding a lot of research. Professor Feng, I think he's got, like, the last round, I think they got about $3 million in funding, to advance the delivery, the vectors.
Yeah.
And so I've got an interview with Monica on Friday morning as well. Mm-hmm. She's the CEO of this trust. But all the research that they're doing, it's, there's the delivery and then there's the gene, and all of it's kind of, it benefits everybody.
Yeah, of course.
And I know they're working on a lot of non-viral therapies that are kind of coming up in the next couple of years. Mm-hmm. So yeah, it's just
different delivery mechanism.
Yeah. So very very exciting times.
I think once the kind of proof of concept is demonstrated, and people can feel confident they can develop these kind of mini versions of genes and create a model where they don't overexpress. Mm-hmm. Right, you can do it with anything, theoretically. It's amazing. Yeah, it's amazing. And what used to be so impossible to target, the central nervous system issues, we might kind of like open the floodgates. Yeah.
So how does so the research that you guys do here and the programs that you do here, the experimental therapeutics program, mm-hmm. How does that change with the things that are going on with gene therapy?
Yeah, exactly. So, we have an amazing team, but we don't have 1 million people, and so we have to be selective about which studies we do, just for bandwidth reasons.
Yeah.
And so, we had so many ideas of our own that we were really excited about. For a long time, we stopped collaborating with industry because there was a lot of these sort of Me Too medicines and small molecules that we didn't feel were as compelling as some of the stuff that we were working on for specific genetic forms of autism. And so now I think we have to sort of recalibrate and make sure we have room to do other gene therapy studies. And I just got approached by another company today to be a site, and
for PMS,
not for PMS, for a different rare genetic disorder, but similar strategy, similar phenotype in some ways, and I want to make sure we can be part of all that because it's exciting and it's important to be able to contribute. So, I could see us not in the not so distant future just being involved only in disease modifying types of therapeutics, mm-hmm. whether it's gene therapies or ASOs or and moving away from a small molecule. Mm-hmm. I think there'll always be a role for small molecules potentially in terms of like symptomatic improvement, but clinical trials are very, very hard and they take a really long time, especially in rare diseases, to kind of recruit new people and collect data and analyze data. So you have to be pretty thoughtful about what you participate in. But yeah, I mean, to your question, I think we'll probably shift our portfolio and focus on things that are really high impact. Yeah,
That's really exciting.
yeah, it's really exciting. I mean, we spent a lot of time collecting data for natural history studies, for phenotyping studies, for biomarker discovery studies, and that's really interesting. But what we were most excited about is obviously developing new treatments. That's at least what our center is entirely focused on. Whether it's people that are working with big data or mouse modeling or even epidemiology, everyone's kind of focused on how do we treat the patients, how do we affect the patients. And this is the ultimate, right? So yeah, we'll make sure everyone do all these studies.
Just thinking of 10 years from now, when Rett syndrome and Phelan-McDermid, these things are just nobody will really have it anymore.
Yeah, that's the hope. I mean, there's certainly a lot of genetic disorders that have been cured. So absolutely.
And then what does that mean for the healthcare system?
Yeah.
And regardless of whether it's a private insurance thing or a public insurance system, what that means in terms of cost savings and
just to shift the whole model from treating illness and focusing on waiting for diseases to emerge and then trying to treat them symptomatically versus wellness and prevention.
We're growing up in a hell of a time.
Yeah, I mean it's mind-boggling. It's actually mind-boggling. Yeah, what used to feel like science fiction is now like stuff that we're actively working on.
Was this I guess 10 years? Did you have any kind of inkling that this would happen in?
Yeah, so in science, there's always the new greatest and latest technology that everybody's excited about that's going to answer all the questions, and it could be chromosomal microarrays or neuroimaging or big data. And so, yeah, I think the idea of gene therapy was among these ideas that everyone talked about as being the next frontier. And we are on the precipice of it, and it has obviously worked in many conditions. So, like I said, I'm optimistic, but I'm still a little bit cautious.
Sure, yeah, sure. Are you able to speak to the work that you do with Jaguar?
Yeah. I mean, there are limitations, but I just speak generally.
So, in what capacity are you guys working together?
So I started with Jaguar a few years ago when they were preparing to go into their first human trial. And I was just, helping them better understand the phenotype, thinking through design, thinking through outcome measures for the clinical trial. We have a data use agreement with them, so the developmental synaptopathies consortium, where I'm the lead for the Phelan-McDermid syndrome network. We've collected natural history study data for 10 years now. So that was really valuable to Jaguar. In so much as it's going to actually serve as a control, right? Because, typically you need to do placebo-controlled studies. Can't really do that with a gene therapy. But when you know what the natural history looks like over what we have now is 200 patients, you get a sense of what things look like without gene therapy. So that was really important to Jaguar. And now I'm going to be the PI as one of the sites, which is Mount Sinai. And so we will do in the next, hopefully 3 months, the first, dose essentially of the gene therapy.
So the first phase or phase 1/2 is ages 2 to 9.
First phase is ages 2 to 9. And then we'll sort of make sure it's safe, make sure there's signals of clinical improvement, and then, likely expand the ages.
So can you take me through what that's going to be like? So phase 1/2, they're going to dose their first patient, and is it they wait a couple of months, making sure, for the safety, and then for the second, if everything's fine, then second patient, they'll kind of modify the dosing, and they do that for the first, I guess, 6 or so patients.
Yep.
And they follow them for a period of time, I guess, so maybe 18 months or something like that before they kind of get, move on to a phase 3 or...
Yeah, so I think there'll be 2 cohorts basically at 2 different doses. And the first cohort will be the lower dose. And they'll be monitoring it for at least 6 months to make sure that it's safe. And then, likely kind of enrolling more patients and then eventually expanding the age range. The original study was designed for 5 years of monitoring.
Okay.
But that's not to say that they're going to wait 5 years before they start to expand the inclusion criteria.
Right.
Yeah. So I think the goal for the company is to get this to as many people as possible, and as quickly as possible. But just, obviously with the caveat that we need to be absolutely certain it's safe.
Right. Right. And basically it's they get the treatment, which is an intracerebral
ventricular ICV.
ICV. I'll just say ICV.
Yeah, it's a bit of a mouthful. Yeah. It's a single dose ICV, and followed by, I guess, then like they get like ABA therapy, or right. So everyone's going to have some therapeutic program. It'll include behavioral therapy, speech therapy, occupational therapy, physical therapy. It's going to be relatively standardized, or at least the goals will be pretty clear. Because these things don't exist in a vacuum, right? It's got to be the intention is sort of like the reinforced learning.
And will the patients have that? Like, is there going to be like a control? Are they going to have that therapy prior to the?
So we have a sense of what their therapies are prior, based on the natural history study. Yeah. So we'll feel like kind of get some idea of like how people improve with certain types of regimens.
Amazing.
Yeah, it's amazing, and the Jaguar has been very, very committed to SHANK3, and they're moving things as fast as they can. And there's a huge amount of optimism, and it's very exciting.
So, like, if things go well, like an FDA approved therapy is looking at like 5 years, 6 years, or quicker.
I mean,
I know they're on the fast track, investigational new drug and all that stuff. So,
I always get asked that timeline question, and I think it's a little challenging to answer. It depends on so many different factors.
Mm-hmm.
But one would hope within 5 years we have a very clear sense of whether this is going to be, approved or not. If not already approved, if it's successful.
And by the time this is out there, how many other therapies are going to be in trial?
It's clear. I mean, it's just unbelievable.
Yeah.
Because again, it's this sort of proof of concept about how to deliver genes to single gene disorders.
You mentioned biomarkers. When we were in Boston at the CureSHANK symposium, there was a presenter, and I forget who she was. She was talking about imaging biomarkers for Phelan-McDermid syndrome, about recognition of items, I think, or when someone with Phelan-McDermid syndrome sees an item, with someone without Phelan-McDermid syndrome, when they see an item in imaging, they can see item that the person recognizes.
Mm-hmm.
With Phelan-McDermid syndrome, that's not there.
Mm-hmm.
Do you remember that at all? Or
so I think you're talking about April 11.
Okay.
We've tried many different biomarkers, and imaging has been among them. I think imaging is a little challenging, and there's different kinds of imaging, right? You have structural imaging that people do clinically to make sure there's not like a big cyst or some sort of mass, and then there's functional imaging where you look at what parts of the brain activate. We've done studies looking at sort of the brain's response to speech and non-speech sounds. Mm-hmm. We've seen that actually within Phelan-McDermid syndrome, kids are activating the appropriate area of the brain in passive listening tasks in the scanner. But they're sedated in the scanner. They have to be. And sedation and the procedure and the cost has not been ideal. And it's very hard to do that in a kind of clinical setting, obviously. Mm-hmm. So we've shifted a lot our focus to electrophysiology, just using basically EEG markers. Mm-hmm. That's, I think, a lot of what April was presenting. And so ideally there'll be a way to sort of stratify groups of people with Phelan-McDermid syndrome based on different biological markers that are objective, rather than just relying on clinicians reporting or parent reporting. That will help us understand who might respond to different kinds of treatments. And in Phelan-McDermid syndrome, there's definitely kind of patterns of the waveforms that are produced when you listen to sounds, or when you see visual stimuli that are very clearly different than healthy controls. So those are all potentially can serve as biomarkers, and we're testing some of them, and Jaguar Group will be testing some of them in the context of their trial.
How many sites are they doing? And so this is one of the sites. Are they doing more than one site, or is it all?
Yeah, in all likelihood, there'll be sites across the developmental synaptopathies consortium and perhaps even beyond that. But at the start, it'll be us and Rush, and then likely Boston Children's and Stanford. And so it'll start to expand. We want to make sure that patients have access. It's burdensome, right? You need to be within, I think, a half an hour of the site for the first 30 days or so.
Oh, okay.
Yeah, because there'll be a lot of pretty intense monitoring.
Okay.
We want to make sure people have access in case there's any problems. But that necessitates lots of sites to make sure that everyone can sort of get there easily without upending their whole life.
Right.
Yeah.
Do you know? So, are there any barriers? I mean, aside from geography, but are there any barriers to like families from Canada or families from Europe? Coming and being
Relocating.
For a month or whatever it is to be part of the trial?
We've talked about Europe. We're not going to have European sites, but if families from Europe are able to relocate, I'm sure.
Is there anything that's, are there any, legal hurdles for somebody that's not a U.S. citizen to take part in a trial?
So the costs associated with the trial should be paid for by the sponsor.
Right.
So you don't need to have local health insurance. But whether or not it'll be feasible for families to kind of relocate, because again, it's going to be a 5-year study that they're committed to.
Right.
Even if approvals come sooner, we want to make sure we monitor people for 5 years. I know that we won't have international sites, but whether we're going to prevent families that are traveling from abroad, I don't actually know the answer to that. Good question. One consideration is that we want to make sure we're not only selecting families with means. Sure. Because the further you travel, the more expensive it is.
Really?
Yeah. So I think, we end up sort of drawing circles around sites and kind of selecting on that basis. But at some point, as a rare disease, and especially when we're focused on such young kids at the beginning, I do imagine families will be traveling pretty long distance. Yeah.
Yeah. So you mentioned ChatGPT.
Yeah.
You're starting to play with it personally.
Yeah.
Have you guys started using it at all in terms of your research?
So less clinically, although we have one new assistant professor, Shlomi Becker, who's working on an AI model to try to predict based on lots and lots of data who might have Phelan-McDermid syndrome. I mean, you talk about your odyssey trying to get the genetic diagnosis. There's really no what we call pathognomonic feature of Phelan-McDermid syndrome, right? If you come into a clinician's office, there's no one specific thing that they're going to see and that's likely to be Phelan-McDermid syndrome. Let's get you tested. But there may be. It's just probably like a agglomeration of lots of different features. So we had to figure that out, and that's something that AI and machine learning can eventually do. So people are excited about it. Obviously, people are working on it. In our clinical research right now, we're not actively using it, but we will be. We have to be. Like I was saying before about how every few years is a new kind of mad buzzword and research that you have to start to think about. And it was genetics and imaging and big data, and now it's AI. So, yeah.
That brings us to the end of our conversation with Dr. Alexander Kolevzon. A huge thank you to him for sharing his insights, his research, and his unwavering commitment to families living with rare genetic conditions. His work is helping shape a future filled with possibility, a future we call Generation Hope. If you found this conversation as meaningful as we did, make sure to subscribe to the channel so you don't miss upcoming interviews with the doctors, scientists, and researchers driving the next wave of breakthroughs in gene therapy. And to stay up to date on all the latest gene therapy news from around the world, visit us at generationhope.co. Stay connected, stay hopeful, and we'll see you in the next one.